Good morning, everyone. My name is Wee Joo Cheng. I'm from the Yong Loo Lin School of Medicine in the National University of Singapore. It is my pleasure to be the moderator for this first panel discussion session at this year's Global Young Scientists Summit. In this session, we will be discussing the potential use of very exciting gene editing technologies, particularly the CRISPR system, for personalized medicine. As a clinician, a hematologist that treats patients with blood disorders, particularly blood cancers, I can fully appreciate how blunt some of our treatment tools are today that we have, with a lot of non-specific activity, off-target activity, and really not addressing the key issues that underlies many of our diseases, which are genetic based, whether they are germline mutation or somatic mutation, as in the case of cancer. So, as a physician, I was particularly excited about the emergence of these technologies and what the future may hold with the use of these technologies in treating diseases. Joining us in our discussion today are two Nobel laureates. Well, in fact, this is the first time I've ever spoken to one, let alone two, in my life. So this is a real honor for me, and preeminent experts in the field. It is my honor to introduce Professor Thomas Cech from the University of Colorado Boulder from the United States of America. He was awarded the 1989 Nobel Prize for Chemistry for his discovery of the catalytic function of unprocessed RNA. He's also well known for his work with telomerase and the telomere. The discovery that he made provided some new tools for gene technology. Our second panelist is Professor Jennifer Deltner, who actually, as I found out, was Prof. Cech's graduate student. So the connection here is extremely strong indeed. And Professor Deltner is the professor in the Department of Chemistry and Department of Molecular and Cell Biology at the University of California, Berkeley. She's best known, and I think for those who are all in the call today would clearly know that she did pioneering work in CRISPR gene editing, for which she was awarded the 2020 Nobel Prize in Chemistry, along with Emmanuel Charpentier. So who better to discuss gene editing and personalized medicine with us today than both Prof. Cech and Prof. Deltner. Today, we will be discussing the following areas. We'll start off with a short description of how CRISPR works. And then we will discuss the potential medical use, what could potentially be the low-hanging fruit, the initial targets for medicine and treatment. What are the ongoing technical issues, safety concerns, and then whether there are any ethical issues that need to be addressed or not. The flow for this session would be in a form of a fireside chat. So I'll be talking to the panelists. But we invite our participants to send in their questions through the chat function throughout the entire 1 hour of this session. And we have staff that will identify the interesting questions that we may pose to our panelists for further discussion, either during the discussion itself or towards the end of the session. So do not wait till the end to send in your question. Do send it in as and when you have question and the discussion is going on. We also hope that the session will be a bit more interactive. So for those questions that are selected, we will try to let the questioner come online to ask the question personally. So our staff will actually open up the video and the voice for you to ask the question. So without much further ado, we'll commence the session. So good evening, Jennifer and Tom. Thank you for joining us for this session. And I think maybe we'll start with a more general question on, what is CRISPR? How does it really work? And why is it potentially better than previous gene editing technologies, say, for example, like TALENs and so on? And I think maybe I'll address that to start with to Jennifer to kick us off. Thank you.
Well, thank you, Wee Joo. And I just want to say good morning to everyone in Singapore. It's really an honor for me to be here, especially with both of you and with Tom Cech to discuss this. And, so CRISPR is in biology, it's a bacterial immune system. It's a way that bacteria find and destroy viruses, and they do it using an RNA-guided mechanism. And the reason everybody's really excited about it is that it's become a common technology now for genome editing, using it to change DNA sequences in cells in a precise way. And so it provides the kind of precision that people have been wanting for manipulating genomes. And it provides that precision in a very accessible, simple to use kind of package that makes it easier to use than earlier genome editing technologies. So that's really what's happened over the last 8 years, is that it's just sort of taken off as a tool that lots of people now are using globally, really, to do gene manipulation in plants and animals and human cells.
Thanks very much, Jennifer. Tom, you want to add anything to that?
I would love to add maybe a couple of things. Just in terms of the technology, I find it important to realize that the RNA-guided CRISPR system does the cut of the DNA. But then what happens afterwards is done by the human cells repair machinery. And so we have a little less control maybe over what the human cell does than we did with the cutting itself. So that may come up again in the discussion. The other thing that I think we could mention at the very beginning is that my laboratory and Jennifer's laboratory and many other laboratories using this as a tool in the laboratory with human cells, it's they're very accessible when they're growing on a petri dish, right? You have good access to the cells. So even though this is a fantastically robust machinery, getting the delivery issue for human disease is going to be easier in some cases and very daunting if you need to deliver it to the middle of the brain, for example. So maybe the blood system, the hematopoietic system, will be one of the most accessible tissues in the human body. So those are just a couple of additions to Jennifer's nice introduction.
Great. Thank you very much. And you know, as you mentioned, you have started to mention about, you know, the potential application in medicine. Perhaps I would, you know, get your view as well on what do you think are the real kind of medical potential? Is it mainly to deal with genetic disease, or there are sort of broader application beyond that to start with? Maybe Jennifer, you can start with that again.
Well, genetic disease is clearly one exciting application of CRISPR, and as Tom said, using it in the blood supply, in the blood system, to change the genes that are responsible for causing things like sickle cell disease, that's already underway, and other blood disorders is one of the early applications of CRISPR. And then there's, of course, many other opportunities to correct disease-causing mutations if the challenge of delivery can be addressed, again, as Tom said. But I would also point out that one of the things that's really interesting about CRISPR is that it goes well beyond human medicine. So it's also being widely used now in agriculture. It's actually being used widely in microbial systems, microbiomes, and doing microbial editing there. And actually, in the face of the current pandemic, there's a very active effort in a number of labs and companies to use it as a diagnostic tool as well. So there's, you know, it's a very versatile kind of system that can be deployed, and it's all kind of fundamentally dependent on the RNA-guided mechanism, which makes me and Tom very happy, I'm sure, because we're both kind of RNA lovers from way back.
Absolutely.
Tom, do you have anything to add to that?
No, not really. I mean, I think that one, again, I'm not nearly as much an expert on this as Jennifer is, but one disease state that I've am very excited about that's just a terrible disease is muscular dystrophy, which is a genetic disease, and the children usually die when they're teenagers. I knew one who passed away when she was like 18, and this normally male disease, but this is an area where Eric Olson at UT Southwestern in Texas in the United States has made some just remarkable progress, interestingly, by changing the RNA splicing pattern of this large gene. So I think that human ingenuity and scientific, creativity are so powerful that we will find ways to overcome some of these challenges. And the fact that we're starting out, due to the discovery of Jennifer Doudna and Emmanuel Charpentier, with such a robust tool that has such high activity, really makes it worth trying to make a medical miracle out of some of these.
And maybe as a non-expert, although I do work in the cancer field, and we do know that, say in cancer, when we talk about genetic defects, it's actually a whole range of different kinds of abnormalities. From sequence mutation to translocation to deletions and so on. Is this technology potentially applicable to correct and reverse a whole spectrum of different abnormalities, including, say, the creation of new super enhancers? Say, for example, can we actually delete the super enhancer and hence reduce the overexpression of certain gene? Is it as broad as that, or actually its main application is towards certain types of genetic abnormalities and correction of that only? Jennifer, maybe?
Well, it certainly can be used for that kind of manipulation. I think in the cancer field, and I'm curious about what Tom thinks about this, but my view of that right now is that probably CRISPR will be useful primarily as a way of manipulating the immune system, immune cells, to get them to attack cancer. I think it's probably going to be very challenging to use CRISPR directly in tumor cells, because you'd have to get every one, and I think that's going to be hard.
That was exactly what I was going to say. That you could think, okay, well, you've got a mutant KRAS, which is driving cancer. We know that CRISPR can be used very efficiently to remove a gene. Okay. But now the problem is let's say you get it up to some remarkable 90% efficiency. Well, that may be of very little value, because the other 10% of the cells will quickly take over the population. So there are some limitations that are just part of the biology. The biology gives some limitations.
Mm-hmm.
Yeah, great. I think maybe we can move on to the next issue, which we have started to touch on, actually, which is what are some of the technological hurdles and challenges. And for this, we have some questions from the audience, which I think are already immediately relevant. We have started to talk about the use of CRISPR in complex genetic diseases like maybe cancer. And I think there is a question from Christopher Ortiza. Can we get him up online next? About the use of CRISPR in, say, germline genetic conditions that may be driven by two genes. So, can you target more than a single gene at the same time? But I'll get him to come online and address the question. Are we able to do that? Christopher, if you are online, could you click on share? And we will be able to get you up on the screen to ask your question. Great. I think he's joining us soon. The technology is working, hopefully.
I'm impressed.
Yes. Hi, Christopher. Very good morning to you. Yeah, you have the airtime to ask your question to the two experts. Go ahead, please.
Thank you. It's an honor to see two Nobel Prize winners in chemistry in one screen. And actually, the question I want to ask is, how are we going to deal with genetic diseases that are associated with multiple genes, not just by a single gene, using CRISPR?
Yeah, well, I can take a stab at that, I guess. And I guess the short answer is it'll be harder, you know, because it becomes a kind of a combinatorial problem where you want to target multiple genes, and if you need to have editing occurring at those multiple genes in every cell, then you know you can imagine that it becomes more challenging, certainly, than if you're going after a single edit. That being said, there's some really interesting, I think, and very encouraging work that's being done both in animal cells, maybe in some human cells, but certainly in pigs, in mice, where multiple editing, multiple edits are being introduced quite efficiently. And also in plants, amazingly, in plants, there's a lot of work, and people refer to it as multiplexed editing, where you can use a series of guide RNAs that direct the Cas9 protein to different genes at once in the cell and to induce editing. So I think it's sort of at what I would call the kind of the bleeding edge of the technology. It's a direction that a lot of labs are working to improve efficiencies, but still needs further development.
Okay. Thank you, Professor Deltner.
Thanks, Christopher. So, we're moving into the area of technological hurdles, I guess. And actually, Tom had earlier mentioned that in his, well, at least the first thing that he mentioned in his mind was potential issue with delivery of CRISPR to, especially in the human system where it's so complex and there's, a lot of hurdles in the way to getting somewhere to a target site. We have a question from our audience in the relevant area. Philip Boscovich, we're trying to get him online to ask his question. And in fact, he has two questions. Okay, and he's coming in now, soon. Okay.
Tom, you should take this one.
Sure.
Philip, good morning to you. Thanks for joining us. I think it's good evening to you actually on your side, or good time. It looks very late on your side. Please go ahead and ask your question.
Yeah, Cambridge now is night. So the question that I was keen on, and I think the one that links also to the Professor Lang talk, can we actually make CRISPR-Cas systems smaller, or can we actually make gene editing systems smaller so it's easier to be delivered? And I think that's one of the key problems, because Cas9 is huge, complex machinery. But can we somehow, by engineering it, now with biotechnology and the chemistry, can we make it simpler? Thank you.
Yes, thank you, Philip. I think that's certainly something that progress can be made. One way to get the Cas9 into a cell, of course, is to put in the gene for it, so that the gene would then be expressed to make the protein. Since you have to put in the guide RNA anyway, which is a nucleic acid, one might be able to co-package the guide RNA with a gene for the Cas9. It's just one of many ideas. I'm not trying, I have no patent in this area. I'm not trying to sell anything here, but that's one possibility. I might just say that the recent success of the Moderna and BioNTech vaccines that are fairly large messenger RNAs being delivered by lipid nanoparticles gives me some hope, that delivering nucleic acids, which has just been a very tough road for many decades, that there are some new breakthroughs in the engineering of these lipid nanoparticles. And, you know, I'm pretty optimistic.
Philip, I think you have a second question. You want to ask that as well about choosing,
Yeah, I think that might be relevant to the previous, but CRISPR-Cas look, at least for me, as you mentioned, not only for monogenetic diseases, but can we actually for tandem repeat associated disorders actually solve that just by actually removing extra DNA?
Like trinucleotide repeats?
Yeah.
Yeah, Jennifer, you want to take that one?
Sure. Yeah. So I think that's a really interesting question. And in our own lab, you know, one of the very first applications we had for CRISPR in my academic lab was to target the gene for Huntington. That, as you may know, when it has an extension of triplet repeats, that actually induces disease, Huntington's disease. And so what the strategy that was taken by a former postdoc, Brett Stahl, who was doing this work, was to look for single nucleotide polymorphisms that you could actually target on either end of the repetitive, you know, sort of the large repeat element, to essentially excise it from the gene. And he, you know, had some success doing that in a mouse model of Huntington's. So that's the kind of strategy that, you know, one could take with that kind of disorder, where you could, you know, and kind of relevant to the previous question about making multiple edits or multiple cuts in the genome. You can actually use it to literally, you know, chop out a segment of DNA and remove it from the genome.
Yes, I like your question, Philip, because in general, it's easier with CRISPR to cut something out than to have to repair. It's more efficient to cut something out than it is to have to repair, like a point mutation. And so I think that your idea of using this with trinucleotide repeats, or ALS has the C9orf72 hexanucleotide repeat, you know, that's pretty promising area. Maybe you should work on it.
Great. Thank you very much, Philip, for your question. Thanks for joining us. I've got another question from Siti Oslan, which I think it's an interesting technical question, maybe helpful to many working in the field at a more junior stage, is that it's the choice of technology, really. I mean, if we have one particular gene to edit from a yeast genome, of course, this is in yeast and not in human disease, but I think it's nevertheless relevant. Based on your experience, which method will be faster and more efficient, using homologous recombination or CRISPR? And in this aspect, do we have, I guess, what is the appetite for FDA and others for doing trials using CRISPR technology in terms of funding and regulatory approval? So two slightly different questions, but I'm asking on behalf of Siti, who is unable to join us online. Maybe Jennifer, again, down for you can go with a technical question, and maybe I ask Thomas to address the funding and the regulatory appetite question, if you may. Yep.
So I think the technical question referred to using it in yeast.
Yeah.
Is that correct?
Right.
Yeah. And so for those that have worked with yeast, and for me it's been a long time, but I did once do a few experiments in yeast way back when I was a graduate student. But you know, yeast are remarkably good at doing homologous recombination on their own, even without CRISPR. And with CRISPR, it can be, you know, maybe even more efficient, but I think it's, you know, it's an organism that's already kind of predisposed to do that kind of genetic manipulation. So I don't know which is better, you know, for a particular experiment that you might be doing. I think in general, you might find that CRISPR makes it even more efficient than a process that is already relatively, you know, facile in yeast. That would be my answer there. But I would defer to, you know, Tom, you may you may think differently. I don't know. You have more experience, I guess, in working in yeast.
No, I totally agree with you, Jennifer. I was amazed when I found out that people were using CRISPR in yeast, because as the student very nicely pointed out, you have yeast is one of the few eukaryotic systems where you had this ability to do gene editing through this very facile homologous recombination system. So I think if in your lab, if you're in a yeast lab and you're totally conversant and successful with traditional yeast molecular genetics, I don't think you need to use CRISPR.
Great. And Tom, do you have any thoughts about the current appetite for CRISPR in terms of trials and so on? And is the FDA kind of likely to approve these technology?
I would like to defer to Jennifer because she's thought about 100 times more about this than I have.
Sure thing.
Well, you know, I think I have a couple of thoughts about that. First of all, I think there's a lot of interest on the part of regulatory committees like the FDA to figure out how they're going to appropriately regulate a gene editing tool like CRISPR. And, you know, because we're talking about personalized medicine here, I think it's appropriate, you know, it would be worth mentioning that on the one hand, CRISPR is great as a personal tool because the whole, you know, the beauty of CRISPR is that it's programmable. So you can literally personalize it to a particular genome, which is great. But the flip side of that is that, you know, taken to an extreme, it would mean that, you know, CRISPR for each of us would be different. And so how would you ever do clinical trials where you are testing something in an individual that then has to be changed in somebody else? So that's clearly not going to, you know, that won't be possible to do a clinical trial for every kind of iteration of CRISPR. So I think one thing that's happening right now is looking for diseases where there's a way to make CRISPR as a tool that is more generally applicable. So, and a great example is going back to what we said earlier for sickle cell disease, where it's possible many people have a similar mutation, and so you can use the same form of CRISPR in principle to treat different people. And so there you could imagine doing trials, and in fact they're ongoing. But it gets harder when we think about really rare diseases where, you know, on the one hand it's great to have a personal tool, but then how do you think about testing it appropriately?
Very, very fair point. Actually, I think that's always the challenge for any sort of personalized medicine approach is, you know, how do you assess it? We have a next person who's going to ask the question live, Emmanuel Galang. He has an interesting question, which I must admit that I'm not really familiar with. So Emmanuel, you are live with us. Go ahead with your question.
Hi guys, greetings from the University of the Philippines, Los Baños. It's my pleasure and my honor to be speaking on behalf of my university here. I have a question with the recent discoveries on Detector, which is intended to detect specific sequences. Were there recent applications or have there been studies which have established the tandem use of Detector as well as CRISPR to address a specific medical condition?
Okay, well, I can take a stab at that. So I think with Detector, you're referring to a form of CRISPR that's being used for a diagnostic.
Yes.
Yeah. Yeah. And so it is CRISPR. It's just using it in a fashion where it's used for detection rather than for genome editing. And so one of the really nice things about CRISPR as a diagnostic is that it can be programmed with multiple guide RNAs to look for different sequences in one reaction. So let's take the example of the coronavirus, the SARS-CoV-2 virus, where people are discovering mutated forms of the virus that are cropping up and can sometimes be missed in standard PCR type assays, where a tool like CRISPR, again, in principle, could be used to capture all the sequences that represent different forms of the SARS-CoV-2 virus because of this ability to do detection of multiple sequences at once.
And if a new variant shows up, you can add that to your list in a week, and you can have it up and going again.
Yeah.
Thank you very much, you guys. That sounds very promising, specifically with the COVID-19 pandemic that we have. Thank you so much from the University of the Philippines.
Thank
Very
you.
nice to meet you.
Thank you. Great. Thanks very much, Manuel. We have rather a lot of people interested in this kind of area. So we have I guess we'll take the last question in this segment before we move on. And this is from Mark Carrascal, who is also going to ask the question online. So hopefully Mark will pop on soon. There you go. Yeah, Mark. Welcome, Mark. Thank you for joining us. You're live now.
Hi.
Please go ahead with your question to
Yes,
the
can
professor.
you hear me?
Yes, very well.
Yes. Hello, everyone. Well, first of all, I'm Mark from the Philippines. I'm actually a clinical microbiologist, and just before asking my question, I would like to, state that I'm very happy to be part of this session, especially with time. So my question is really side, because as a clinical microbiologist, we're really into that tenet that these technologies should be accessible to the people who this technology should really serve. So my question is on what are the current strategies in making CRISPR technology a more point-of-care type of technology in terms of diagnostics? And second, what are the current strategies in making CRISPR more accessible to the scientific community? Because speaking from a country with no access to this high-end technologies of the moment, we are really relying on these strategies on making CRISPR a more accessible technology to everyone. Thank you very much.
Well, I guess both of you probably could address that question from your own perspective, right? Yep. Maybe Tom, go ahead.
Maybe Jennifer could handle the diagnostic point of care one, and then I'll do the more accessible to the scientific community.
Sure. Well, the point of care idea is being developed by a number of groups actually right now who are using CRISPR to detect viral RNA in samples like saliva and nasal swabs. Making it into a true point of care test, I can tell you from personal experience, is challenging, not so much because of CRISPR itself, but it has more to do with just, creating a test that's really easy to use, fast, kind of bulletproof in terms of, being very, very easy. You don't need special training or instructions to do it. And where we can, one challenge we have right now, honestly, at Berkeley is just dealing with our environmental health and safety people who don't want, saliva or nasal swab samples that could have virus in them going into a point of care test. So, we're trying to figure out how to manage that. But I think in principle, it's a solvable problem. And whether we'll see a really robust point of care test in the next few months, I think it's possible. But I'm really sort of thinking down the line about future pandemic preparedness. And as Tom said earlier, one of the things that's great about CRISPR is that if a new virus pops up or a new variant of the current virus, we can quite quickly create a format for CRISPR that will detect it. So I think that's the real utility there. And once a format for dealing with all these other sort of logistical issues that can be solved, the CRISPR technology fundamentally is going to be very useful for point of care testing.
So Mark, thank you for your question about the accessibility of the research technology to the scientific community. We understand very well that we are in a privileged situation in the United States, in Singapore, and in many other countries in terms of the resources we have to do science. And we are sorry that those are not more worldwide, broadly available. I will say that the CRISPR technology is not particularly expensive or inaccessible. Any laboratory in your country that has the ability to grow human cells on petri dishes and to transfect plasmids into them already, this is no added expense. Of course, if you don't have those fundamental tools available, then that's a huge barrier to entry to the field. I will just remember when 6 or 7 years ago, when I was first interested in using this in our lab, I called up Jennifer and I said, we need to collaborate with you. We need some help. And she paused for a minute and she said, well, I would be glad to help, but it's really easy. She said, just try it. It will work. And so I had a student from China who was the first person at our university to get it to work, and she was able to get it to work quite quickly. So it is, there are some barriers to entry for third world countries and other less developed countries, but certainly this is not an especially high standard or especially expensive.
Can I just add one thing to that? So Mark, I'm curious whether you have access to adgene.org. Do you know this organization, Adgene?
Yes, I think I've heard of it, and some of my colleagues here.
Yeah. So it's a nonprofit organization set up in Massachusetts that distributes research reagents, including CRISPR, just at cost. So they don't, it's pretty inexpensive to get it. So that would be one way. And obviously you can always reach out to us if there are, constructs that you can't get a hold of some other way. We're always happy to send them out.
Jennifer, how many Cas9 plasmids has Adgene sent out around the world?
Oh my gosh, I've lost track. I mean, if you add up all the different labs that are, placing constructs there and then Adgene distributing them, it's got to be, I don't know, many tens of thousands.
Yeah, I think it might be 100,000 or something like that.
Yeah, I wouldn't be surprised. Yeah.
Great, thanks very much.
Thank you very much. That's very wonderful.
Thank you, Mark.
Yes, thank you very much.
Bye-bye. I think that was a great session, actually. We'll move on now to some questions related to safety. There were a couple of them somewhat related, but anyway, I think it would be great if we can get the participants to ask those questions online. The first will be, I wouldn't even try to pronounce his or her name, right? But Yosipol, who we are trying to get online next. She should. There you go.
Hello.
Oh, hi. Good morning, good evening to you. Please go ahead and ask your question.
Hi, Jennifer and Thomas. My great pleasure to talk to you today. I have a short question regarding a clinical trial, and I think Jennifer trying to raise the campaign of a global pause. For some example of disease like sickle cell, that it's about the disorder in genetic information. So it's only way to treat these diseases by CRISPR-Cas at the moment. But now it seems that we have the global pause to stop a clinical trial in humans. So my question is, what would you like to prove for avoiding unintended consequence of the CRISPR in humans? I mean, for example, like sickle cell, we don't have any chance to treat those people instead of CRISPR, but now we stop this trial. That means a lot of people now are suffering instead of we can cure them immediately. So what would you like to prove to make this global issues move forward?
Jennifer, you want me to say something first, and then you can
Yeah,
add in?
sure.
Well, I think that that you have to go through safety tests. You usually would try this first in a mouse model of the disease, and then perhaps what in the example I gave previously, there was which is muscular dystrophy, which is a genetic disease. There turned out to be a dog model in the UK, and the researchers were able to cure this muscular dystrophy in these dogs. And so then at the same time, they're looking for side effects, they're looking for safety issues. And I think that you have to go through those steps. You can't just start with humans, because it's too risky. So I appreciate that you would like to speed this up, and we all would like to speed it up. and we can speed it up to a certain extent, but ultimately you don't want to, if you do something that injures many people, then it sets the whole time course of the thing back it gets a very bad reputation then. So safety has to be considered at every step. And usually the animal models, although not a guarantee of safety, are a good intermediate step. Maybe Jennifer has other ideas.
Right, yeah. I think, I think about this a lot because I hear what you're saying, and, people are suffering, and you want to, I think all of us as scientists, we want to see our work benefiting people as quickly as possible. But as Tom said, it's almost, it is worse to have something go forward prematurely and then harm people. So, and maybe, harm the whole field. And, Tom and I both remember, years ago when gene therapy was just coming to the fore, and there was a really unfortunate event in a clinical trial in the US that led to someone's death. And that really set back the whole field of gene therapy for at least a decade. Mm. So, I think we want to avoid that, obviously, here. But you're right. It's always a struggle, to figure out, you want things to move quickly, but not too quickly. And I think that it's just essential that, everyone conducting these trials just continue to be really vigilant about safety, and that people like me and many others who are doing more fundamental work are continuing to, make sure that the technology is working correctly, that it's not, it's not creating off-target edits, that it's, that it's ensuring the kind of changes that are going to be beneficial. And then, it's up to clinicians then to make sure that that's, when it actually gets introduced into people, that it's done in a safe manner.
Yep. Thank you very much.
Thank you, Jacopo, for your question.
Thank you.
We have another question which I thought is quite relevant from Amit Ghosh. Again, he's going to join us online to ask his question. And this relate to, because we said that medicine, even targeted therapy, may have off-target effect, and this is related to whether CRISPR may also have off-target effect and how specific the editing is in human. Is Amit joining us? Okay, he's coming on soon. Mm-hmm. Great. Hi, Amit. Good morning.
Good morning.
Go ahead with your question to the professors.
So it's pretty straightforward. First of all, it's an honor to ask questions in this session, and very nice to meet you all. And so my question is what are the health issues which are associated with CRISPR? What are its risks? So can it accidentally shut down something important, or even cut down something in the untargeted tissue rather than the place we intended to go in the first place?
Tom, do you want to speak to that?
Well, I'll just briefly say, and then Jennifer can add on, off-target cutting, off-target editing are unavoidable at some level. You can minimize them. Ultimately, though, there is that possibility. You'd be pretty unlucky if the off-target event shut down an important gene, but it could happen. And ultimately, that's why we have to do clinical trials, right, to see what the safety of something is. And I work for sometimes for Merck, the pharmaceutical company, and they say the best drug to take is no drug at all, right? Because every drug has some negative side effects in some people. And so of course, it's always a question of risk-benefit ratio. If you're going to die from a disease, if it's deadly, then this risk of an off-target effect is probably something you would dismiss rather quickly. On the other hand, if it's just sort of an annoying disease that you have that can be treated by other means, then it might not be tolerable to to do CRISPR editing. Jennifer, you want to add to that?
Yeah, I know. You said it very well. I agree completely. I think it's all about, the risk-benefit ratio, and for severe disease, you're going to be more willing to accept a risk than not. I would just add that, I think, and you said this already, Tom, that there's probably never going to be a therapy of any kind that will have zero risk.
Yeah.
So we're always going to be dealing with some level of risk, and with CRISPR, one of those risks is some kind of off-target editing, and so we work on ways to reduce that, and and to try to get it to a point where it's really considered safe enough, given the benefits, to use it there as a therapy.
Yeah. Great. Totally agree.
Thanks very much, Amit. Thank you.
Thank you so much.
Thank you, Amit.
We'll move to a next question, which I thought also is quite relevant and interesting. We're focused a lot on the genome so far in our discussion, but this question next from Maya from the University of Auckland, he's going to ask the question online, is related to, whether, CRISPR could actually induce certain autoimmunity or some immune reaction that may be potentially harmful to the patient that's being treated. So let's get Maya on to ask the question. Great. Ooh. Maya, can you turn on your video?
Maya is very shy.
Well, he looks good though. You should show his face. If that was indeed his photo. You're on mute as well. I think, I'm not sure, is he on mute?
Hi, can you hear me?
Okay, it's okay. Yeah, Maya, why don't you go ahead and ask your question? Please.
Sure. Hi, it's an honor to be here in this panel discussion. Exciting topics. So yeah, my question was more about immunity. I'm not from this background. I don't have a strong background in CRISPR and genome editing, but I'm quite interested to know whether for potential CRISPR treatments for genetic disorders, is there any concern regarding any immune responses, against CRISPR? Any thought about that?
Yes, I think it. So people are worried about that with the mRNA vaccines right now against coronavirus, right? So putting in foreign RNAs into the human body, because there is an innate immune system that we have that is poised to be set off by RNA, because RNA usually means virus coming in, right? So from the outside. So it is a concern, and maybe the Moderna and BioNTech vaccines will give us some idea about whether we need to worry about the RNA component of CRISPR.
And actually, I thought you were going to say, Tom, that it seems like another angle to that is the question about natural proteins that are made in cells. So let's take the example of muscular dystrophy that you brought up before. You could imagine that for a patient who is affected by muscular dystrophy, where their cells are producing one form of this dystrophin protein, when you edit that to make a kind of a corrected form, that might look like a foreign protein. And so could that induce an immune response? And I know that's something that people like Eric Olson, who are working in the field, are thinking about.
So, Mayar, I think the answer to your question is we always worry.
Yes. Right.
Great. Thank you so much. Thank you so much for your time.
Yeah, thanks, Maya. I think a lot of the questions in this segment relating to safety really have to be sort of observed and pan out in the human trials, right? I mean, a lot of these are theoretical in nature at the present time. So I think the next phase of CRISPR, as we do more clinical studies, will definitely be able to learn more about that. I think we are doing quite nicely on time to move on to the very last segment, and we already have lined up 3 questions from the audience regarding ethical aspects of CRISPR. I know this sometimes can be a bit uncomfortable, but I think it is nevertheless something that is on top of people's mind. So the first person that we will have asked the question live is John Guzman. Yeah, he's joining us online now. John, please go ahead with your question.
Wait a minute, I'm having some problems. Do you?
No, we can hear
We
you.
can hear you.
Oh, wait a minute. Can you hear me?
John, okay, we can't see you. We can hear you, and we see a nice photo of you. I presume
All
that's
right.
how you look like.
All right, all right, all right. So it's actually, first of all, I would like to thank everyone. Thank you for the opportunity of asking the question. Actually, I'm going to ask this question in behalf of some of my students, and I'd like to ask now. So since we're talking about personalized medicine, how do we address the worries of the people regarding this technology being used in quote unquote playing God, since there are issues concerning the development of what they call the designer organisms, especially now that biohacking or the DIY or the citizen or the DIY science is on the rise.
I would say that Jennifer Doudna has been a real leader internationally in thinking about the ethics of the CRISPR genome editing. So maybe she would like to start on this one.
Well, it's a great question, John, and thank you for asking. I think it's something that all of us working in the CRISPR field think about, because it is quite a profound thing to have a technology that allows precise alteration of a genome in any organism, but including in humans. And you know, if it were to be utilized in a human embryo, that creates a heritable change that makes a change that can be transmitted to future generations. So you know, it really is a technology that requires, I think, careful thought, careful regulation. My view of it right now is that I think the benefits of using it to treat diseases in individuals is so exciting and so profound that we certainly want to see that field moving forward as quickly as possible. And it looks like we might have lost John. I hope he's still able to listen. But you know, I think that, and I think many people in the international scientific community agree that that it's not an appropriate technology to use right now in human embryos for many reasons. So you know, the vast majority of work that's going on around the world, both in academic labs and in companies. Oh, there he is. Hi, John.
Hi.
Is really focused on treating disease in an individual, not to create a heritable change. And there I would argue that CRISPR is, then very much like any other type of therapy that you might utilize. It affects a person and you want to be sure it's safe and effective in that individual, but it doesn't affect anybody else.
All right. Thank you. Thank you very much for that. Actually, let me just share with you that actually this question, when I reported or when I taught my students about this CRISPR, especially in your recently awarded Nobel Prize, they kept on asking that if we allow these people, are we trying to engage them in doing science, or do we have this sort of gatekeeping in allowing people with no, or how do we say this, formalized training in science in doing this CRISPR? Yeah. So thank you. Thank you very much for that.
Thank you, John.
All right.
We have another question from Yusang Dorji, I think. Again, on the area of ethics. I will ask the question on her behalf because we couldn't get a response from her. She is coming on. Oh, great. Well done. And I think this is about, again, similar to the previous question, the issue of how it may affect not just the human, but other species as well. Yusang, please go ahead with your question.
Hello, everyone. First of all, thank you so much for this amazing opportunity to talk in front of 2 Nobel Prize winners. So my question was related to the CRISPR technology. This provides a lot of opportunities, but there are also some ethical concerns, not only for humans, but it will also affect other species. So are there any ways that we can address these issues, ethical issues related to CRISPR?
Oh, she disappeared. Well, thank you, Yusong, for that question. And I agree. I think, again, we have to be very thoughtful about how a technology like CRISPR is used in the environment. One example of that we haven't discussed yet in this session, but is certainly a topic that comes up frequently in the CRISPR community, is using CRISPR to create changes that can be spread quickly through a population, for example, in mosquitoes. And we call that a gene drive. And, in principle, that is a use of CRISPR that could, cause unexpected or unintended environmental impacts if it, kind of got sort of out of control. On the other hand, it could have a very positive impact on human health and sort of public health if it were possible to use it to control, a disease that's spread by mosquitoes. So, again, one has to kind of balance, risk and benefit in that type of application. And again, I have to say that at least right now, I've been quite encouraged by the response globally of the scientific community. I think people have really recognized that this is not a tool to be deployed, without caution. So there are a lot of groups that are working in these areas who are, doing research, but doing it in a way that really takes into account the kind of safety measures that we need to have to ensure that we don't see unintended consequences of CRISPR.
Tom, you want to add anything to that?
No, I thought that was a very good response. Thank you, Jennifer.
Cool. Great. So we'll move on to the next question from Eric Onos. Again, in this general area of ethical concern, Eric will join us. Eric, please go ahead with your question.
Hello, everyone. I'm Eric from University of the Philippines. First of all, I'm very thankful for this opportunity. So in 2019, Professor He Jiankui from China was criticized for his controversial research on twin babies, CRISPR babies, in whom he tried to edit the CCR5 gene hopefully for HIV prevention. So there are legal and bioethical concerns that were raised by this research. Can I ask the panelists to comment on this issue? Also, do we have global efforts to prevent this controversial research? And ultimately, do we think that CRISPR technology can be ultimately used for editing out genomes in human embryos? Thank you.
Well, I'm actually curious, so I said a few things about that in response to a previous question, but I'm actually curious about Tom's opinion about this because, what I've kind of sought to do, especially with respect to the He Jiankui situation and, possible future use of CRISPR in human embryos, has been to really try to engage the international scientific community through the scientific societies and through other organizations that have weighed in on kind of the appropriate ways to proceed with the technology. And I'm curious, Tom, whether you feel that is an effective strategy. Do you think there are other things that could or should be doing to address this issue?
Not really. I think that again, the distinction that is important here is between germline editing, which would be inherited by subsequent generations, and treatment of somatic tissue or stem cells within an individual, which would not be inherited. And so it makes total sense to start with the somatic treatment and to treat the individual and not the germline, knowing, of course, that if it's a serious genetic disease, what you really would like to do would be to eliminate it from the germline. But it's sort of a stepwise thing, and there are so many possible misuses of the CRISPR in the germline editing for enhancement of various traits rather than curing diseases. I frankly, I don't know if I should say this in public, but I think this will come, you know, at some point, the germline editing. I think it's just the history of science that if something can help people, you can't keep it bottled up for forever. But I think that for the coming, you know, decade, maybe more than that, that we should avoid, we should have a prohibition on those experiments until we understand more about safety and efficacy.
Right. Thank you so much.
Thank you very much, Eric. Thank you. I think we're doing fantastic for time. We have a couple more minutes. We have some fantastic question. I think it has gone better than I expected. The technology actually did work very well for us. So maybe I have, I will ask a couple of, in fact, related question. Just one last question that in screening through the questions, there were a couple of colleagues that have asked this question. And I'll ask this question of Tom, because he's, as he says, an enthusiast for the CRISPR technology. And so he should be unbiased in that sense that he can look at the CRISPR in comparison to other technology in an unbiased manner. So there were a few questioners that asked about what are the, you know, with the advent of CRISPR, are there still useful roles for other technology like TALENs, zinc finger nucleases, Cre recombinases, or even the RNAi, you know, short hairpins, the kind of technology in science, or everyone should be just using CRISPR because it's so much better, more accurate, cheap. There's no reason why you shouldn't use it. So Tom, what is your take on that? Do you use other technology in your lab still, or are you just using CRISPR?
Well, I think that I may disappoint some of my friends who invented some of these other technologies, but I think that the zinc finger nucleases and the TALENs are history at this point. They are so tedious and expensive and less efficient than the CRISPR. I think they're sort of gone. Maybe that's too extreme. The microRNAs, short hairpin RNAs, I think have, we still use them. I must say that they almost are guaranteed to have off-target effects, often quite serious ones, which people don't really think about. They don't want to think about them. So I'm afraid that that is a bit of a primitive technology, in my opinion. And I mean, after all, biologically, think about it. The microRNAs are meant to control a whole pathway, a whole series of genes. So having this sort of broad specificity rather than precise specificity is critical to their biology. So I think that they're somewhat on the way out as well. Although they will still be used. The Cre recombinase, we actually use that in conjunction with CRISPR sometimes. So it's a tool that has some use for recombining out a segment of DNA in the genome. Although again, it's not without its attendant problems.
Great, thanks very much. Jennifer, anything to add to that?
Well, I guess just real quick, I would add that you know, I think that you know, we use RNAi methods in my lab now still, you know, but for particular purposes. Like if we're really interested in not manipulating the gene itself, but manipulating the RNA transcript of the gene for some reason, then, you know, that is the way to do it. So I think you just have to look at, you know, what it is you're trying to do, and then use the tool that makes sense to do it.
Yep, great. Thank you very much. Thank you both for your excellent, you know, answers to all the and patience actually with the technology, and all our participants with your question and addressing these very important issues with us today. We've come to the end of the session. So enjoy the rest of the meeting and hope to see you all soon. Thank you very much.
Thank you.
Thank you, everybody. Thank you, Weiju. Thank you, Tom.
Thank you, students from around the world.
And students. Yes, thank you.